explain both of them, but initially I would concentrate on complement. Now, what does this particular word mean? Actually, complement, it means to complete the activity of antibodies. Now, what is antigen and antibody is known, right? Anything foreign to our body that is introduced either in the form of bacteria, virus, or any other pathogen, or sometimes certain biomolecules. So these are treated as antigen against which our immune system that reacts and produces certain effector molecules. Molecules particularly, that lymphocytes, they get activated against them. But before that also, our immune system has got certain receptors that can identify the foreign objects. These might be in the innate hand, and these receptors can recognize the pathogen associated molecular patterns. These are specifically present on pathogen surface and against which our immune competent cells can react. And lymphocytes particularly, they are the prime players of the adaptive immune system. And if I just focus on two broad categories of lymphocytes, B cells and T cells, I would like to ask the students if they know about the different aspects of B cell and T cell. Do you people know that what are the basic types of lymphocytes that are present in mammalian immune system? Students, if you can answer. Hope they are capable of listening to the queries. Students, anybody of you? I think Ritesh sir, they are listening to, right?
Yes, 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 they are. Students,
Okay.
please respond. When sir is asking some question, please respond. Ruhi,
Yeah, it is
yeah,
better to
Garima,
have an interactive session. If I just only
right, right.
talk, you might not be feeling very interested in this particular talk, right? So if it is an interactive one, then it becomes enjoyable to everyone. Students, can anyone respond to whether you know the basic classification of lymphocytes? What are the different lymphocytes present in mammalian immune system? Anybody?
Yes, sir.
Okay,
Yes, sir.
okay. Thank you, Ruhi, for responding. So the B cells and T cells, right? So the
Yes,
B
sir.
cells, you know that they produce the antibodies, particularly not in their B cell form. When they get activated, they transform themselves into plasma cells and then secrete antibodies. These antibodies come into the system and they can bind to the pathogens, for example, bacteria. But on the surface of pathogen, now what happens to the pathogen? Nothing. The pathogen still remains well because the size of the antibodies are too tiny in respect to the size of the bacteria. If something foreign binds to the bacteria, it doesn't uh, create a lot of problem to the bacteria. But it is certain other aspects present in our system that can assist the antibody to render some toxic effect on the bacteria. Is it clear?
Yes, sir.
Okay, fine. Now, this aspect was being dealt by several scientists. You know, the development of immunology, the modern immunology is not very old, right? As a subject, immunology is very recent. In the last, say, around 100 years only, we know about the different aspects of immune system, right? Particularly, the humoral hand, the cell-mediated hand, these things. Now, we were talking about the activity rendered by antibodies. They alone cannot destroy the pathogen, such as bacteria, right? And if I just present this slide, can you see this scientist? The figure of this scientist, Jules Bordet. Have you heard the name of this scientist? He actually was working in Pasteur Institute in Paris. Okay, and he first established the role of these complementing factors. Complementing factors are the factors that assist the antibodies to render the toxic effect to the pathogen. I hope this is clear to everyone. Students, can you please respond to?
Yes, sir.
Okay, very fine. So you see, Jules Bodick, what did he do? Before that also it was known that antibodies cannot do alone because adding antibodies to the pathogen didn't kill the bacteria or the pathogen, right? So it was known, but what are the factors? This was unknown, right? So like scientists like Paul Ehrlich, they also knew that certain complementing thing is there that assists the antibodies to kill the bacteria. But it was Bordet who finally discovered and critically explained the role of these factors in killing bacteria, and for which in 1919 Bordet was given the Nobel Prize also. You know, immunology as a subject in biology has clinched the most of Nobel prizes, right? There are several scientists who clinched Nobel Prize, and Bordet was one of them, one of the very first scientists from immunology who claimed the Nobel Prize. And what was the experiment? It was a very easy one, you know. Can you see this figure? This is a figure representing a very easy experiment. In this figure, you can see a container that is having the antibodies generated against certain bacteria. It was Vibrio cholerae. Okay. And if these antibodies are present in serum, you know, antiserum. If this serum is known as antiserum. If a model animal is given an inoculum of certain pathogens. Suppose a bacteria is introduced into the blood circulation system of the rodent or the animal. The animal was sheep here. So you see the body of the animal develops activated lymphocytes, out of which B cells are also activated, and B cells transform themselves into the plasma cells, and plasma cells start secreting antibodies, and these antibodies can be harvested from the serum. You know, blood can be collected from peripheral blood vessels, and if this blood is kept to clot, the serum that is harvested afterwards contains the antibodies that are generated against the pathogen. The pathogen here is Vibrio cholerae. Okay. Now, what happens if this serum is mixed with the bacteria? Now, if bacteria is mixed with this antiserum, the bacteria get killed. So you can see this lyses the bacteria. But Bordet found that if this antiserum is heated for some time, up to say around 56 degrees centigrade, the serum, this antiserum, loses its capability to kill the bacteria. Is it understood? Is it clear to everybody? Please
Yes.
respond. Okay, fine. So all of us now understand that if antiserum is mixed directly with the bacteria, bacteria gets lysed. If antiserum after heating for some time up to 56 degrees centigrade, the antiserum loses its capability to kill the bacteria. Now what could be the factor? If antibody was a factor, antibody was not lost because antibodies were intact, even intact in at 56 degrees centigrade temperature. Now, how it was proved? When fresh serum that was not collected from a inoculated sheep, that was a normal sheep. It was not injected with the Vibrio cholerae. So obviously it is not anti-serum, rather only serum. When a fresh serum was mixed with the inactivated antiserum, it regained its capability to kill the bacteria. So is it understood? Antiserum kills the bacteria. Antiserum heated, then it loses its capability to kill the bacteria. Antiserum heated But when fresh serum, not anti-serum, fresh serum from the similar individual was mixed with this heat inactivated anti-serum, it regains its capability to kill the bacteria. So what was the picture? That picture clearly says that there is certain factor present in normal serum, not it is a inoculated, means not a activated, immunized serum, right? Normal serum contains certain factor that assists the bacteria, that assists the antibodies to kill bacteria. Is it understood?
Yes, sir.
Yes, sir.
So what were these factors? Later on, these factors were established by several other scientists, including Bodet, that these were actually proteins and they were thermolabile proteins. What does it mean, thermolabile? It means that even heating up to say around 50 or 60 degrees centigrade, these proteins lose their general structure, and obviously if they lose their general structure, then also loses their activity. So these are thermolabile proteins. I hope all of you understood this experiment. It was a very nice experiment performed by Jules Portet. And later on, he also characterized certain proteins. Nowadays, we know that lot more number of proteins are there, around 50 different types of proteins that are also expressed, not only expressed on plasma membrane, but also they are secreted form. Okay, so these proteins, they actually assist antibodies, not only antibodies, later on different other aspects of the complement protein were discovered. So initially, as it was established as an assisting factor for the antibodies, it was known as complement that completes the activity of antibodies. I hope all of you understood this.
Yes, sir.
Is there
Yes,
any
sir.
query regarding the experiment? If you have any query or confusion, you can ask. Shall I move forward?
Yes, sir.
Okay, fine. So these proteins, they constitute around 15% of the globin fraction of proteins present in our serum. So obviously it is not less. There are many different proteins, out of which a big chunk of proteins, they constitute the proteins that represent the complement factors, and they are around different 50 different types of proteins. Now, how do we name them? These proteins can be named according to the C for complement and numericals like 1, 2, 3, 4, 5, 6, and so on. Out of which that C1 molecule that is constituted, it's a pentameric protein actually. One molecule of C1Q along with two molecules each of C1R and C1S, they together form the C1 molecule. This is the very initial molecule that actually begins the classical pathway of complement activation. Now, what is this pathway classical? That we will discuss in detail. But initially, we should know that how the complement proteins are recognized. So these are proteins, and these are actually present in the form of zymogen in the serum. Zymogen means these are not present in the active form, but can produce the active form when they are cleaved. So when a protein is cleaved, it is the fractions are known as part A and part B. Like if we can cleave the C4, the C4 will produce C4A and C4B. Generally, B is the larger fragment and A is the smaller fragment. I hope all of you understood this one.
Yes, sir.
Okay. So alternative pathway of proteins are there, and then classical pathway of proteins are there. In the classical pathway, they are named as C and then followed by numericals. But in the alternative pathway, they are generally recognized as factors or identified by single letters, different letters like factor B, factor F. factor H, factor I, and so on. I hope all of you understood this one. Now, complement receptors are there in different cell types. Complement are proteins and their receptors are also present in different cell types. Okay, and these complement receptors are recognized or named using the CD format. CD, like CD 16 molecule is there, CD 21 molecule is there, CD 19 molecule is there, and so on. Many different complement receptors are present on different cell types. I hope all of you understood the naming pattern. Is there any query?
Excuse me, sir.
Yes,
Sir, just now I think I missed something. You said proteins present in the form of zymogen.
yes.
And then after that, when protein is heated, and there is two part of proteins, part A
No,
and part B.
not after heating. When the activation process starts, it can break down into the active form and the inactive cap type of thing. As if a bottle is opened, then only we can use the content of the bottle, right? So that cap is removed. Cap is removed in the form of A, and B is the active form. This is the general pattern. I hope you understood that proteins are broken down, then only their active portion is opened, right? So they are present in the inactive form. Now you can, if you have any queries.
So the activated part is the zymogen.
No, it is normal form that is present in the serum. It is the inactive form. Okay, zymogen that generates the zyme or enzyme. Okay, that generates zymogen. It is not generated in the inactive form. Okay, it's a pro-enzyme type of thing. I think you know that pro-enzyme, when the cap part is removed, then only it becomes enzyme. Is it understood?
Yes. Yes, sir. Thank you, sir.
Okay. So shall I move forward?
Yes, sir. Sure, sir.
So, what are the roles of these proteins? If we can brief the role, there are different very important roles played by these proteins. Opsonization is one of them. What is opsonization? Opsonization is a thing that enables the immunocompetent cells or phagocytic cells particularly to phagocytose the pathogen. As you can see here, these are the pseudopods. I hope you can locate the cursor on the screen. Can you locate it on your screen? I'm just pointing out the pseudopode that are generated by the phagocyte to capture a bacteria. Can you locate it? Students, are you capable of seeing the cursor?
No, sir.
Okay, let me just use that one.
তিলকদা এই স্ক্রিন
হ্যাঁ, আমি একটা পয়েন্টার নিয়ে নিচ্ছি।
ওই
Now can you
জুলস
see
এর
that?
ওখানে আটকে আছে কিন্তু, মানে তুমি শিফট করছো?
হ্যাঁ, আমি শিফট করে যাচ্ছি।
না না, it is stuck at that Jules slide, Dr. Jules
ওকে,
slide.
ওকে, 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 ওকে। আমি আরেকবার একটু শেয়ার করছি।
Yes, I changed now.
হ্যাঁ, এখন আসছে।
এখন আসছে না? তাহলে এইভাবেই আমি দেখাই নাকি?
এখন
সবাই
দেখা
দেখতে
যাচ্ছে।
পাচ্ছে তো? আচ্ছা, ফাইন, ঠিক আছে। So as you can see, this represents the role of complement proteins. So complement proteins, if they are bound to the surface of the bacteria, they enable the phagocytes to capture it. So this is the process of opsonization. And then chemotaxis. Chemotaxis is a thing whereby when these complement proteins, their smaller parts, actually the A portion more often, they help the other immunocompetent cells to move towards them in a chemical gradient. So this process is known as chemotaxis. And then another thing, this is the target cell lysis. When complement activation is completed, it forms MAC. What is MAC? MAC is membrane attack complex. Now, when MAC is formed on the surface of the pathogen or the target cell, sometimes our own cells can become harmful, right? These cells are also killed by complement and other component of the immune system. So, these MAC can be formed on the target cell membrane to lyse the target cell or the pathogen. Is it understood? Students, have you understood the three functions?
Yes, sir. Yes, sir.
Okay, fine. So, in gist, this complement protein defends against pyogenic bacterial infections. The normal chance pathogens, okay, very common bacterial infections are inhibited by these complement proteins, and they actually bridge the gap between the innate and adaptive immunity. How do they do it? We'll discuss on it later on. And they also can assist in disposing the immune complex from the system. What is immune complex? Actually, we know about the antigen and antibody, right? So antibodies actually cannot do any harm to the antibodies of the pathogen. They can only bind on the surface, right? And antigen and antibody complexes are formed. These complexes are sometimes very large, and in the experimental system, we often term them as precipitin. Okay, so these are large macromolecules composed of many different, not different, many of the antigen and the antibody molecules. Okay, together they form complexes. Now, what will happen if these complexes are go on accumulating in our system? Our system will be clogged, right? Our system will be clogged, blood vessels, smaller, particularly finer blood vessels will be clogged. So the system removes these debris frequently. It is a continuous process by which our immune system, particularly the phagocytes, they can clear these antigen antibody complexes from the system, right? So this process is known as, disposing of the immunogenic debris. Okay, so all these very vital functions are carried out by complement system. Actually, they can get activated in three different ways. One of them is classical pathway, another is a lectin pathway, another one is alternative pathway. As I stated, that complement, they actually bridge the gap between the innate and adaptive immunity, right? Innate immunity means our body's immune system is having a pre-formed antigen recognizing molecule, right? It acts against the antigens. It is already there. It doesn't need to get activated. But the adaptive system, where particularly the lymphocytes get activated against antigens, right? Whatever antigen comes into the body, there might be some form of the lymphocytes, because the lymphocytes are having the variable type of receptor present on their surface. So might be there might be some of the lymphocytes that can be able to bind to those newer antigens, right? So this is the adaptive form of immunity, and these antigen antibody complexes are formed. And these antigen antibody complexes, as I mentioned, that antibodies cannot harm actually the bacteria, but later on they can activate the complement system, right? So antigen antibody complexes that can lead to the activation of complement, and complement actually can create pores or holes on the bacterial surface, or even uh, give the cues to other phagocytic cells. Now, phagocytic cells in a chemotactic way, they can run to the area where antigen antibody complexes are formed, and they can capture the pathogen. I hope it is understood that how the antigen antibody complexes can create the classical pathway of complement activation. Now, in between these two, classical pathway mediated by the adaptive immune system, alternative pathway is mediated by the innate immune system. What is innate immune system? In the alternative pathway, actually you know that the certain components of the complement system, like the C3b molecule, it readily gets cleaved. Whenever certain pathogen molecules come in our body, they these complement molecules are just like mines. You know, in the border area, what happens? Mines are put so that whenever enemy comes in the surface, in our surface, they will be killed. So similarly, these complement proteins, they can easily break whenever pathogen comes in and can create the alternative pathway of complement activation and particularly leading to the killing of these pathogens, right? So this doesn't initiate with the antigen antibody complex, but readily the complement proteins get cleaved and finally MAC is formed. So this is the innate immune mediated pathway. I hope all of you understood this one. The difference between the classical and alternative pathway. Is it understood?
Yes, sir.
Yes,
Yes.
sir.
Okay, very nice. So in between these two distinct pathways, there is another pathway known as the lectin pathway. Lectin pathway, it begins in a similar way like the alternative pathway, but with a definite molecule known as the lectin. Lectin, like manoj binding lectin is there, MBL. Okay, these manoj residues are often present on bacteria and certain other pathogens. So these lectin, MBL lectin are sensors present and these, can initiate the activation of complement, right? So this is another pathway that initiates with the lectin molecule. So in these three different pathways, complement gets activated. So I hope you understood this. This was explained like the following caption. Here you will see classical pathway. So it begins with the immune complexes, right? It can act against apoptotic cells, viruses, gram-negative bacteria. Okay, and the lectin pathway that begins with the managed groups. Right, these are the terminal ends present on the terminal ends of the microbes. An alternative pathway that can begin against bacteria, fungi, viruses, tumor cells, etc. But all of these have common endpoints. As you can see, the formation of C3 convertage. C3 is a complement protein. C3 convertage, that means that further C3 molecule will be broken. So that can convert the C3 molecule. That itself is produced by C3. So you can see here in the classical and lectin pathway, the C3 convertage is C4b2A. But in the alternative pathway, it is known as the C3BBB. Now, what these factors are, we will later on understand. Let us begin with this classical pathway. As I mentioned earlier, that C1 molecule is a pentamer, right? Composed of two of the C2, CR molecules and CS molecules and one CQ molecule. Can you see that umbrella-like thing here in the center, blue colored? Can you see that?
Yes, sir.
Yes, sir.
Okay, so these complex is actually, it is having six different arms, right? Each of the arm is composed of A, B, C. These are the detailed structure. Okay, we don't need to discuss about the detailed structure right now. But this molecule, umbrella-like molecule, is the C1Q molecule. Okay, and here, dumbbell-shaped structures are there. Two of these CR and two of the CAs. So altogether, this is known as the C1Q R2S2. Okay, this molecule actually is the C1 molecule. Now, how is it formed? It is formed actually like this. Can you see this? This is the target cell membrane. Okay, in this figure, you can see the target cell membrane. Can you locate the C1Q molecule in this figure? Anyone? Either it might be present on the left or right corner. Can you tell me where is it? I hope all of you can see that throughout, starting from the left corner, the C1Q molecule has been represented. Can you locate that one? The blue colored one?
Yes, sir.
Yes, sir.
Okay. So let me just enlarge this figure. Can you see this right now?
Yes, sir.
Okay, very nice. So you can see that this is the target cell membrane, and here on the surface of membrane certain proteins are there. These are the antigens, the yellowish golden color thing. These are the antigens against which antibodies. Can you look at the antibodies? I think I must show it with a pointer.
Yes,
Can
sir.
you look at the pointer?
Yes, sir.
Yes,
So
sir.
this is the antibody. Can you see that Y-shaped antibody molecules? Y-shaped antibody molecules I am pointing out with the cursor.
Yes, sir.
Yes,
Okay.
sir.
So these antigen antibody complexes are formed. Now what happens when these antigen antibody complexes are formed? The C1q molecule that is activated, it actually binds uh, to the the constant region, the Fc portion. If you are just acquainted with, if you know that the structure of antibody, the antibody molecules, they have the variable region and the constant region, right? So these are the constant region, also known as sometimes as Fc region. So the Fc region actually binds to the heads of this C1 molecule, and the C1 molecule gets activated. When it gets activated, it brings in two of the Cr molecule and Cs molecule. So they create complexes, right? So C1q R2H2 is formed. When it is formed, it can actually break down the C4 molecule. I am pointing it out, the green color one. Can you see that one?
Yes, sir.
Okay, so this C4 molecule is broken down. The smaller portion is released as the form C4A, and the bigger portion that is retained. So you can see that C4b. Now C4b can bind to the surface of the pathogen. So it can bind to the membrane. Why C4b can bind to the membrane, we'll discuss later on. Now C4b can bind to the membrane, and later on it can activate the C2A. So C4b now brings in C2, and C2 is broken down into C2A and C2b. In these actually form, the C2A is the larger one, not the larger one, is the active one. This is actually a alteration of the rule, right? So the complex that is formed is actually known as C4b2A. Is it understood
Yes, sir.
up to this? So C4b2A, as we discussed that C4b2A is a C3 convertage, as it is written here. C3 convertage means it can break down C3 molecules. Okay, this is an enzyme. Multiple copies of the C3 molecule now can be broken down by the C3 convertage. So when the C3 convertage is activated, C3 molecules are broken. So C3 molecules are broken into C3A and C3B. Can you look at that one? C3B and C3A. So C3B is the larger part, and it binds to the C3 convertage, forming a complex known as the C4B2A3B. What is it known as? It is known as the C4B2AC3B. Okay, C4B2A3B. So this is the C5 convertage now. Okay, C5 convertage that can break down C5 molecule, and C5 molecule is broken down into C5A and C5B. So C5B is the larger fraction, and C5B can now bind to other complement proteins like 6, 7, 8, and these 6, 7, 8 together with the C5b forms a complex known as C5b678. Can you look at that one? So see, this is the C5 convertage. Now C5 convertage breaks down the C5 molecule into C5A and C5B. C5b comes into the point, and C5b can now employ certain other molecules like C6, C7, C8. Can you look at that one? These are written on this picture. C5b 678. Can you look at that one, students?
Yes, sir.
Yes, sir.
Okay, very nice. Now you can see here that certain portions of the C7 and C8 molecule can get engraved on the membrane. This is just like a pump. that pump, not pump, better to say it's a crane like thing, right? It has got a external part, that is the C5B, and this C5B is holded by other portions like 6, 7, 8, right? And this acts like a crane. We know that when a building is constructed, crane can move and can deploy other larger components to a site. Similarly here, the C5B molecule can employ many of the C9 molecules. And this C9 molecule has got a particular feature. You know that C9 molecule has got certain regions. These are all proteins, right? C9 molecules have got certain regions where the hydrophobic amino acids are rich in number. If they have hydrophobic amino acids, they can easily be engraved into the membrane. Okay, now what happens? Many of the C9 molecules are engraved on the site where this C5B678 molecule is there. So what happens? Multiple copies of C9 molecules along with the C5B678 form a MAC. This is the MAC. So what is the formula of MAC? C5B678 poly 9. So these C9 molecules